In this next video, we'll look at the human genome. We'll search for genome browser. We'll go to the UCSC, which stands for University California, Santa Cruz, the place that this is hosted. You'll see that there are many other genomes that you can explore, but it defaults to human. We'll search for the location that contains the gene P53, and we get a great deal number of results, but this is the one that we're interested in. And the first thing you notice here, we're at chromosome 17. The P arm is to the left in this web page, and the Q arm is to the right. This red line indicates our approximate location on 17P. Now the first thing we'll notice is there is a lot of information here. If I mouse over, you'll see that different green sections are lighting up. These are called different tracks. And when I'm first approaching a gene, I like to have a simpler view with fewer tracks. All the different tracks can be turned on or off here, or they have different options of how to show them, either hide, dense, squish, pack, or full. I'll keep that on pack and I'll make a few changes. I'll hide RefSeq, I'll hide publications, I'll hide the human mRNAs, hide the ESTs, hide encode regulation, I'll keep conservation, I'll keep SNPs, and I'll turn off repeat masker. Refresh, and here we have a somewhat simplified view. The first thing I'd like to do is I'd like to zoom out by a factor of 10 to get a better look at our genomic location. We're still in this small area of chromosome 17P. Here we see different splice variants of the protein P53. We see other genes nearby, this WRAP53, ATP1B2, a little further out DNA-H2, and this protein called SBHG. And here we see how the human genome sequence is conserved amongst multiple different species, mouse, dog, elephant, and so on. You can see regions that have genes are a little more highly conserved than regions that are sort of intervening space between some of them, although it's conserved here between these two genes quite well. This particular track is the consensus conservation of 100 different vertebrates. These are individual organisms here. And here we see the locations of SNPs. I'm going to browse out again just another factor of 10 until we start to get a significant portion of 17P. Just want to give you an idea of the scale of the genome. Here's P53 again, moving out by another factor of 10. Again, all the genes that are present in most of 17P. If I go out 100, this will probably, will for surely get me the entire chromosome. It'll take a little bit to load. I'll pause. And it's finally loaded, and as we can see, we were highlighting the entire chromosome 17. These are the base pairs that are present, and here are the genes listed in their order. And as we can see, the chromosome is truly immense. When we look at the conservation, we see there's a little gap here. This would most likely be the centromeric region, which we still have had trouble sequencing and don't know the sequence of. But now I'm going to zoom back in, and now I've zoomed back to a reasonable area here. If we click on the name of the protein, we'll come to a page with some useful information, links to a lot of other databases, information that for which this is a fairly unique resource is microarray expression data that's summarized in two sets of experiments here, top and bottom. The bottom is normal tissue and normal adult tissue, and then here we have embryonic tissue as well as various disease tissue, various cancers. And as we can see, again, red is high, green is low, that in the immune system, in the immature immune system, P53 expression can be pretty high. It's pretty low in the cerebellum here. Remember, p53 is a protein that's made quite frequently but gets destroyed quite frequently also. And so that levels don't usually accumulate that high. And here you see in adult tissue, p53 is not expressed a great deal. I'd like to show you one last track before we quit. I will now go back to the encode regulation track. I'll hit refresh. I've zoomed in here. Here's the p53. Here is the 5' region of p53. You can tell by the arrows. So this is the 3' region here. And what I'm interested in is these transcription factors, which we use the technique CHIP, that are binding around the transcription start site here in for p53. And as we'll see here as we scroll down, there are a large number of proteins. I would say there's probably listed well over 100 of proteins which can influence the expression of p53, which is binding the DNA in the transcription start site region of p53. And this is going to highlight just how complex the regulation of various genes could be. Okay, thank you for listening.